Okay, Chapter 18, Section 1, Part 2. Now we're going to be talking about, all right, you have amino acids that are in the bloodstream, taken, into the liver, hepatocytes, or, they're now throughout the body and other cells have taken them up. What can you do with them? What can you do with those amino acids? Well, first, like I said, you need to do something with those amino groups. Okay. Anytime you change the position or swap an amino group from one molecule to another, you're doing what is referred to as amino transferase, or you also see this referred to as transaminase. You're just moving an amino group either from one carbon to another within that same carbon skeleton, or you're transferring it from one carbon skeleton to another carbon skeleton. Which you're going to use, an amino transferase or transaminase, is going to be cell dependent. Not all the cells have access to the same genes or the same menu of available enzymes for that cell type. So not only is it cell dependent, but it's also going to be, what is the purpose? What metabolic function are you trying to do? Okay. Do you have enough ATP? Do you have high enough levels of NADHs so that you don't need to go to the citric acid cycle to make more ATP? Now you're looking to, repurpose, remake amino acid, build up your amino acid pools, or you're looking to do this, you're looking to do that. All of these things have to come into play. So when you look here, here's an example of amino transferase. You're going to take alpha-ketoglutarate and you're going to turn it into L-glutamate. Alpha-ketoglutarate, remember, one of the steps in the citric acid cycle. So we're going to, change it into glutamate. All you have to do is add an amino group here at carbon 2. But the thing is, where are you going to get the amino group? You don't just have ammonia sitting around in the cytoplasm. Well, what you're going to do is you're going to grab one of those incoming amino acids. You're going to take that amino group off of that incoming amino acid and you're going to transfer it over to carbon 2 of alpha-ketoglutarate, making L-glutamate. In the process, that L-amino acid is now going to become what is referred to as an alpha-keto acid. Alpha-keto acids can then be further processed into other intermediates of either gluconeogenesis or other, citric acid cycle intermediates, or turned back into another amino acid of some sort. This transamination reactions transfer the alpha amino group to the alpha carbon. Okay. Remember, the alpha carbon is going to be the carbon after the terminal carboxyl group. So, alpha amino to the alpha carbon of the alpha-ketoglutarate. They consider this to be pretty much, a net zero. The transfer, the pulling off, the transfer has within it exergonic and endergonic steps, depending upon the intermediates, depending upon how it's done. So they consider this to be roughly a net zero. You think back that slide, not only did it have the transaminase, but it also had what is referred to as PLP, Pyrodoxyl Phosphate. It's a coenzyme, a version of vitamin B6, one of those essential vitamins you have to take. It's a prosthetic group with amino transferases. Remember, these prosthetic groups are going to hold all the intermediates in place. So the intermediates may not be interacting with the R groups of the amino acids lining the active site, they will be interacting with these prosthetic groups. We've run into these prosthetic groups before. Remember some of the some of the enzyme active, some of the enzyme active sites that we've talked about that had biotin in them. Well, the biotin was acting as a prosthetic group. It's holding the players, holding the amino group donor, pulling off the amino. As the enzyme pulls off the amino group, then PLP, biotin, or whatever this prosthetic group would hold that in place. As here comes the substrate. Here it's the alpha ketoglutarate. Holds alpha ketoglutarate in place as the enzyme attaches the amino group to the alpha carbon. You see here is the PLP. PLP, under normal, quote unquote, normal conditions, has a carbonyl group here. But during the transamination step we just talked about, this carbonyl group gets replaced with the, amino group as it holds it and aids in positioning it. So this is one of those times where you do not want the cell, the enzyme / cell does not want the intermediates free floating around. Have this hold everything in place in the active site so that it can, the amino group can be pulled off the donor, attached here as the alpha ketoglutarate gets its carbonyl, gets those bonds reformed so that it can now accept an amino group that's going to be plucked off here and then attach the alpha ketoglutarate to become glutamate. And, we have to keep in mind that the PLP is not free floating in the active site. It is covalently linked through a shift base. And no, I don't remember what shift bases are, so if I don't remember it, I'm not going to expect you to remember it. That's that organic chemistry crap coming up again. Anyways, so it is interacting with our groups of the active site of the transaminase, holding it in place so that it can be positioned correctly to accept the amino group, hold it there while the alpha ketoglutarate is processed and ready to accept that same amino group. So we see the PLP comes into play quite often. This vitamin B6 derivative comes into play in transamination, racemization, decarboxylation, enzymatic steps. And as we look here, we see that the different patterns all seem to have an overarching theme of whatever is being pulled off, it's going to be held by PLP, which is being held in place in the active site of the enzyme so that it can now be plucked off and put on the true target substrate of that enzyme to form the final product. And no, I am not going to go through here, PLP, here's the shift base intermediate, the quinoid intermediate, rearranging, and then the final alpha ketoglutarate getting it. No, you can go through and you can look on that. I'm more into what's the pattern of what's going on in the different ways it can be utilized. So the glutamate, what can we do with glutamate? Well, remember, glutamate can either be recycled or can be broken down. Ketoglutarate, alpha ketoglutarate, pyruvate, or glutamate to glutamine, and use it as part of your amino acid pools. But if glutamate is going to be further broken down, like I said previously, you're going to have to get rid of that nitrogen. Okay. This is a step that's going to be occurring in the mitochondria. Specifically mitochondria in the liver. Okay. Glutamate going to be converted to alpha ketoglutarate by getting rid of this, making ammonia, the ammonia that's going to be passed out of the mitochondria out of the cell, converting it into urea for us, uric acid for birds and reptiles. So how's this going to work? Well, glutamate to alpha ketoglutarate. Alpha ketoglutarate. Wow. Okay. Well, we're going to use what is referred to as L-glutamate dehydrogenase. Okay. It's an oxidative deamination of glutamate to give us ammonia and alpha ketoglutarate. Oxidative deamination. This enzyme's in the mitochondria, and it's going to utilize NAD or NADP. All right, so how's this working? Well, first off, we got to get rid of that hydrogen. Whoops. This hydrogen right there, because that's going to be the one that ends up right there on either NAD or NADP. It's going to form this. It's going to go there. So we get this double bond. Carbon-nitrogen double bond. Okay. The oxidative deamination. This oxygen right here. Woo. This oxygen right here. It's going to end up right there. Bring in water. So our intermediate here, as we see here, is going to, oh, keep losing my pointer. I'm going to have this intermediate. Strip off the electron. Going to end up with this carbon-nitrogen double bond. Back and forth. Boom, boom, boom. And then it's going to push this way when water is present. Glutamate dehydrogenase, pull off that hydrogen, form the double bond, then it's going to split water. Two hydrogens, H2O, two hydrogens, two hydrogens. You're going to end up with what's behind my head, and you look in the handouts, NH4. There's your ammonia. This oxygen is now going to go and bind here with this double bond that now has nothing to bind to. So now you have a carbonyl, alpha ketoglutarate. So you're removing the nitrogen, replacing it with oxygen. Alpha ketoglutarate, remember, is also one of these crossroads. Go one way, citric acid cycle. The other way, gluconeogenesis. Transdeamination. Going to use a bit of amino acid transferase and a little bit of that glutamate dehydrogenase. So you mix all these together, you get this idea of transdeamination. You're going to take an amino group and you're going to pull it off, and you're going to either release it as urea or you're going to move it to somebody else. Our friend glutamate dehydrogenase, this enzyme here, operates at an intersection. Again, which way are things going to go? Because that alpha ketoglutarate can be further oxidized, fuel, making ATP, or take the energy there and all those bonds, reform them as glucose, store it away for later, or put it out into the bloodstream to be shared with other cells, gluconeogenesis. The glutamate dehydrogenase, upregulated, ADP levels are low, or I'm sorry, ADP levels are high, which means our ATP levels low. This also, means that there's low glucose levels, means low sugars going into glycolysis, pyruvate going into the citric acid cycle. So you want to take all the alpha ketoglutarate and push it towards the citric acid cycle. GTP levels are high, means you've got enough pyruvate entering into the citric acid cycle. You have enough alpha ketoglutarate being broken down that you're now making lots of GTP, which are going to be phosphate's going to be transferred to ATP. So you have enough. Well, then this is going to signal negatively. You know what? Take everything, either slow down making it, the alpha ketoglutarate, through the steps we just talked about, or if you can't do that, shift all the alpha ketoglutarate to gluconeogenesis. So you have this L-glutamate. L-glutamate is going to help us get that ammonia out of the cells. You get a bunch of L-glutamate. You're going to phosphorylate it using glutamine synthetase. So you're going to pop off the terminal the terminal gamma phosphate, add it on. Boom. So there you go. 1, 2, 3, 4, 5 carbons. 1, 2, 3, 4, 5 carbons. You've got the amino groups, and now you got a phosphate group there. You phosphorylated, gave yourself an intermediary, gamma glutamyl phosphate. Gamma glutamyl phosphate with the same glutamine synthetase. So here's an intermediate. The same enzyme is going to take that intermediate, going to take some of that free, ammonia that we just made, or as a byproduct of other deaminations, of other steps in amino acid catabolism. Got to get rid of it somehow, so you're going to add it onto the gamma glutamyl phosphate, so you end up with L-glutamine, because remember L-glutamine had the 2 amino groups. So that phosphate gets added on and then gets replaced by the nitrogen functional group, the amino functional group. Glutaminase will then take that, add in water, boom, give you the L-glutamate back and the urea, which is now in the bloodstream. So here's in the cells. This goes into the blood into the liver mitochondria specifically, gets broken down, and then the ammonia gets converted into urea. Out it goes with the waste. Skeletal muscles. Got to remember, our muscles, their metabolisms are going to be a bit different than anything we talk about with hepatocytes or epithelial cells and neuronal cells or whatnot. They're going to go through glycolysis, make pyruvate, and depending upon oxygen levels in the the tissues at any given instant, that pyruvate can either enter into the citric acid cycle or it could be turned into lactate for a short term, and then lactate turned back into pyruvate, and so on. But there's also the breakdown of the proteins. Okay, breakdown of the proteins, you absorb the amino acids go through the liver to the rest of the body to be utilized, either, to build up your own polypeptides or to be, catabolically broken down for the energy. Well, they're breaking down amino acids. That means that there's also going to be a buildup of ammonia. So, we saw on the last slide how the liver takes care of the ammonia, transported out from other cells into the liver via L-glutamate, and all of that, and how then it gets turned into urea and then out with the waste. Well, the muscles use what is referred to as Alanine Amino Transferase. Takes the pyruvate and alanine, or takes pyruvate, turns into alanine by transaminating glutamate. So you see this in the muscles, glucose to pyruvate, pyruvate to alanine. Alanine goes out back into the blood, gets, taken up by the liver. Alanine gets turned into pyruvate, glucose, via gluconeogenesis, back to the blood, boom, boom, boom. So this is separate from the system that we've talked about in chapter 16, 15, 16 of the making, to get NADHs and all that stuff that we can then use for ATP synthase, the oxidative phosphorylation. This is a little side channel for getting the ammonia out of the muscle cells. Take the ammonia, attach it to, glutamine to make glutamate. Pyruvate takes that ammonia off of that. Boom. So the glutamate becomes alpha-ketoglutarate again. That ammonia then goes to alanine. Pyruvate, the ammonia, that amino group goes to pyruvate, which now becomes alanine. Alanine was easily transported out of the cell, goes in the blood. Alanine easily taken up by liver cells. And then we see the reverse. Pull off the amino group from alanine, add it to alpha-ketoglutarate, and you get glutamate. Go through, break down glutamate, you release that ammonia. That ammonia, which is going to turn into urea, and then out with the waste. The pyruvate gets built back up to glucose via gluconeogenesis, out into the bloodstream, taken up by liver, muscle cells, and you go through this process all over again. All of this is just to get rid of that right there that's building up in your muscle cells. The problem is ammonia is toxic, okay? Especially the brain. If it gets into your bloodstream, builds up, it will become damaging. Turns out the ammonia competes with potassium. And that whole sodium-potassium ATPase thing. Our sodium-potassium pump stopped working. Also our sodium-potassium cotransporter, symporter, doesn't like to work in the presence of ammonia. So you stop maintaining the differential across your membranes. The chemiosmosis, it's based on this, will halt. That will be bad for cells.